Okay, I think we could go ahead and get started for our third leg of our symposium today. And I'm very pleased to be able to introduce Dr. José López, who is probably known to all of you, but I'll give you a bit of a bio sketch anyway. José is a full member at the Bloodworks Northwest Research Institute in Seattle and a professor of medicine in the hematology division at the University of Washington, and he also holds adjunct appointments in biochemistry, pathology, and mechanical engineering. So obviously he's in high demand. José's work spans 3 decades and has focused on molecular mechanisms of platelet adhesion, biochemistry of platelet receptors, and von Willebrand factor, and the physiological and medical correlates of all that work. So, José, I'm going to let you share your screen and turn it over to you.
Can you hear me okay?
We can hear you just fine, and we're seeing your slides.
Okay, great. All right, yes, so today I'll talk about the significance of vessel wall adherent von Willebrand factor. But first, I would, see if this is working. Okay, here. I'd really like to say a couple of things about Ed and really to thank Ed. Ed's really an inspiration. I kind of think of Vancouver as the land of the magical Eds with Ed Conway and Ed Prysdale, 2 great people. Ed Conway, in addition to being a great scientist, a wonderful scientist, a coordinator, a raven, I probably most admire his dedication to students and to mentorship. I've observed this every year that I've attended the Earl Davies Symposium, and I just think it's one of the best things that he does. So that's why thank you. And it's an honor to speak in this symposium. So the work that I'm talking about really spans about a couple of decades, and it probably started with a study that I was doing with my colleague Jingfei Dong, in which we were examining the interactions of platelets with activated endothelium. At the time, we had identified an interaction between platelet glycoprotein 1b alpha and p-selectin. And we wanted to see how that worked in endothelial cells. So we took platelets, we washed them, and we perfused them in a flow chamber over activated endothelium. What we observed was not really a lot of interaction with p-selectin, but we observed these structures with these long beads on a string-like structures. Each one of those dots is a platelet. And you can see that these are enormous, that they span 3 microscope fields. We thought this must be von Willebrand factor. And even though we knew about ultra-large von Willebrand factor, this is really ultra-large von Willebrand factor. You can, so for example, this one string contains at least 100 platelets, it's enormous. It's this little bar is 100 microns, and so this is several millimeters long. So we reasoned that this could not be ultra large von Willebrand factor. This had to be something else, and this is kind of how we put it together. That the VWF was being released from the endothelium. Some of it was remaining adherent to the surface of the endothelial cells, but was somehow self-associating in the direction of flow and able to capture platelets. At the time, actually, Xavier Ruggeri's group actually did studies that showed that von Willebrand factor, soluble von Willebrand factor, could associate with surface-bound von Willebrand factor. And in studies that we did with Ying Zheng in bioengineering at UW, we showed that the VWF indeed comes out of the cells and does associate into strands that can actually converge and diverge, and those strands will capture platelets. And it's interesting that that this phenomenon is basically governed by geometry and by hemodynamics. So in this case, with Ying, we made a vessel of this particular geometry here. And you can see that the VWF that comes out of the endothelial cells on the vessel wall, there are no platelets or blood in this vessel, tends to travel through this in the shortest distance between this convoluted vessel. And you can see that it tends to accumulate on the inner corners of this vessel. And that's where you get a lot of thickness. And the other thing that's shown here is that the von Willebrand factor can span the lumen of the vessels, and in fact can span distances up to 500 microns. So the other thing, so we knew by then that shear stress was something that promoted von Willebrand factor. So von Willebrand factor self-association, wall shear stress in particular. But in addition, as in the situation shown here, flow acceleration promotes VWF self-association. Here we have a vessel that's a large diameter, and it enters into a vessel that's smaller diameter. And of course, to maintain flow, the flow has to go faster here. So it has to accelerate in this region. And as it accelerates, it stretches the von Willebrand factor fibers. And essentially what you get is a situation where you plaster that surface with von Willebrand factor. And that has a lot of consequences. If we look at a grid vessel, and we activate it and we perfuse blood through it with labeled platelets, you can see then in the presence of a control antibody, you get some platelet attachment to that surface, but after a certain time, they don't accumulate, and that's shown here. But if you take the same situation, but instead of this control antibody, you add an antibody against ADAMTS13, the VWF cleaving protease, then what you do is you get progressive accumulation of platelets onto that wall, especially in the regions of highest shear stress, and eventual occlusion of the vessel. And you can see that quantified here. And of interest, if you take that ADAMTS13 inhibitory antibody and you add an antibody against platelet glycoprotein 1b alpha, now you get no platelet accumulation. And so we did a lot of studies with that, and we knew that that there was something in the plasma that could prevent VWF self-association, because if we purified VWF past a certain point and kept it in a tube, we would never keep it in that tube for long, because it would go onto the wall, and that wall had infinite capacity, meaning that once you covered the surface of the plastic or whatever that was, that the VWF would continue to self-associate and attach to that wall. And we identified from plasma the apolipoprotein ApoA1, which is the major component of HDL particles. And we did a number of studies, and some of them I'll describe here, in which we looked to see whether or not we could attenuate VWF self-association in these artificial vessels. And here you see that in the absence of ApoA1, we activate that endothelium, we perfuse blood with labeled with an antibody against VWF and with labeled platelets, and you see that there's accumulation of these VWF strands, and where the strands are thickest, you get the most accumulation of platelets. So the extent of self-association is related to the extent of platelet binding. If we first perfused the vessel with our agonist, VWF will be released and there will be some flow in there, so you'll get a little bit of VWF self-association. But when we perfuse the blood, now we perfused with added ApoA1, you see that we markedly blunted both VWF self-association and platelet accumulation. Now this vessel system is beautiful and wonderful, but it's a lot of work. It's difficult to use. It's hard to have high throughput. And so we noted a study by Scott Diamond's group that was published in 2015, in which they used a microfluidic device such as shown here, in which there was a long channel of 1 millimeter that you could flow plasma through, and in the middle of that channel, there would be a pillar right in the middle, which would essentially occlude 50% of the diameter of the channel. He showed that with this device, you could get accumulation of VWF on that pillar and develop strands that essentially had to be the result of VWF attachment first and then self-association. And we indeed were able to replicate that. Here shown a citrated plasma perfused through the channel, and you can see that these strands of von Willebrand factor have accumulated, and of course we did also stain them with antibody, and that is indeed von Willebrand factor. This is anticoagulated blood. If we took that plasma and then added EDTA to it, what we get is we get more accumulation of these strands of von Willebrand factor because the EDTA has completely inhibited the activity of ADAMTS13. If we take this situation, citrated plasma plus EDTA, and then we add HDL, then this is what we get. We still get a few strands, but compared to this is markedly inhibited. So of course, I've already mentioned that one of the roles of ADAMTS13 is to actually prevent VWF from accumulating to those levels. So in the absence of ADAMTS13, you can see this is the string area that we're looking at, that over time you get, this is the accumulation you see in citrated plasma, but if you add this antibody that inhibits ADAMTS13, now you get a much faster initial rate of accumulation and also a much higher total accumulation of von Willebrand factor onto those pillars. So that was a quick summary of a lot of work with HDL. We still continue to try and characterize that. But the question that we wanted to ask is, since HDL is kind of considered as the good cholesterol and cardioprotective, and there's another component called low density lipoprotein that's considered atherogenic, we wanted to see whether or not that did anything. And so here I just show you that when we added to plasma, when we added LDL, and this is, I think, to increase the concentration to within a range that one can see in hyperlipidemic patients. You see that when this is what we got with the plasma by itself. If we increase the HDL concentration, we got no accumulation in this particular case. And if we added LDL, now we get this crazy thing that that looks ugly, that you would never want to accumulate on your blood vessel. And this is just quantifying this again. In the presence of HDL, we got nothing. In just plasma, we get accumulation like this. And in the presence of LDL, we get rapid initial accumulation and then a plateau. Essentially, I think this has been kind of saturation. So here's another assay in which we are shearing plasma. And we shear plasma, I think this is for 90 minutes. And we're measuring how much von Willebrand factor is left in the solution in that plasma. And just in the plasma by itself, shearing for 90 minutes removes all the von Willebrand factor. But if we add 6 micromolar HDL, we get almost complete protection of the VWF. If we have 6 micromolar HDL and then we add increasing concentrations of LDL, you see that there's a progressive drop in the protection by HDL. So really what it has to do with is the ratio between HDL and LDL, and this holds still if we double the concentrations of both. Those are the only two that we've actually studied. Of course, you want to see if the HDL and LDL are binding. You presume that they are, because otherwise, how could they do that? But we wanted to formally test this. In this case, adding labeled HDL, shown here in red, you can see that it's binding along this strand that's inhibited here with LDL, and this is with purified VWF. with labeled LDL, we can see that it's binding all over these strands. So in this case, this strand, I'm calling it a VWF strand, but in addition, it contains a lot of LDL and probably a lot of other things in addition. But this formation of these strands completely depends on the presence of von Willebrand factor. So of course you want to know, does this have anything to do, can you do this also in plasma from patients with hyperlipidemia? And in this case, Sergio Fazio from, he was then at OHSU, he provided some samples from patients with high LDL, and you can see here the LDL concentration in a group of patients. Here are the control patients. And between the two, the HDL concentrations were comparable. And if we look at the initial rate of VWF accumulation on those pillars versus the LDL-HDL ratio, which essentially is the LDL because these two are comparable, you see that as there's a pretty much straight line correlation between the LDL cholesterol and the initial rate of VWF fiber formation. So I just have a few studies that I want to talk about now to kind of at least get to infer something about the potential pathophysiologic significance of this phenomenon that we've been studying. And these are all studies that we've done in collaboration with Jonathan Lindner at OHSU in Portland. Jonathan, many of you know, is a cardiologist who is an expert in cardiovascular imaging, in particular with the use of ultrasound. And not only cardiovascular imaging, but also molecular imaging. So Jonathan is able to make these microbubbles that are lipid filled with gas that the surface, and he can functionalize the surface. In this case, what I'm going to show you are microbubbles that have been functionalized with the active A1 domain of von Willebrand factor, which will bind to platelets, or with the N-terminus of glycoprotein 1b alpha, which will bind to von Willebrand factor. And we looked in an atherosclerosis-prone mouse at whether VWF and platelets were on the vascular surfaces. So this is a double knockout mouse that, when fed a Western style diet will develop atherosclerosis, as shown here by oil red O staining of the aorta. And you can see these red dots, that's at 10 weeks, 20 weeks, and of course, the atherosclerosis progresses. As athero progresses, so does the increase in the presence of platelets on that vascular surface. So very early in these lesions, you can already see platelets. And that also is true for the presence of von Willebrand factor. If we take a 40-week, so this guy, and we look at both the presence of platelets or the presence of VWF, you see that both of them are, there's a lot of both of them on that vessel wall. And if we add ADAMTS13, in fact, within a very short while, those are removed from the vascular surface and essentially goes down to baseline by 1 hour. This ADAMTS13 was recombinant from human. Again, if we look in a variety of mouse lines at the relationship between plaque area and GP1B alpha signal, you see that essentially it's a straight line relationship. So the more plaque you have, the more platelets you have, or perhaps it's the other way around. So does this have functional consequences? Well, first of all, we wanted to show that if we added LDL or if we added VWF, we would get more platelet signal. So here we did an ADAMTS13 knockout mouse. So they're injected with LDL or control or saline intraperitoneally and with VWF intravenously. And you see here's the control signal. And if you add LDL, the signal goes up on the vessel wall for platelets. And if you add VWF, it's even higher. If you add them both together, it looks like it goes higher. Similarly, if you have on a high-fat diet, an LDL receptor knockout with ADAMTS13 deficiency, you see that just the addition of VWF will markedly increase the platelet signal on the wall. And this is just intravenous infusion of the VWF. So what's happening likely is that there's already VWF on the vessel wall. We increase the concentration in the plasma, and the plasma VWF will self-associate with what's on the vessel wall. For the VWF signal, we see exactly the same pattern shown here and here. And as to functional consequences, I'm just going to talk about what happens with microvascular blood flow. As you get accumulation of VWF and platelets on the wall, you get a decline in the blood flow. And you can see that both here and here. Another study that we did together had to do with looking at a clinical observation. That is that when somebody has a myocardial infarction, they are at much higher risk of having a second myocardial infarction within a month or two of that first one. In addition, they are higher risk of developing stroke. So in this case, Jonathan's group, they took a mouse and they ligated a coronary artery, and they looked at all of these adhesion molecules on the vascular surface. And this is at distant sites from the coronaries. And you can see that 3 days after a myocardial infarction, you have expression of, you have platelets on the surface, you have expression of P-selectin, von Willebrand factor, and VCAM1. Of interest, this inflammatory state, which is what I'll call it, persists for at least 3 weeks. So here, when you see there's P-selectin and GP1B alpha on that surface, if we treat these mice with ADAMTS13, you can see that the GP1B alpha is completely removed, but there's persistence of P-selectin, indicating that most of this P-selectin is endothelial, a little bit of it may be from platelets. Of interest too, that this inflammatory state is associated with an increase in the growth rate of the plaque. So these are more or less identical groups of mice, except that one group has been subjected to the MI protocol and the other has not. And you can see that the plaque at distant sites progresses much faster in those that had myocardial infarction. And just over here on the right, you can see this is intravital microscopy with labeled platelets. And this is in the mesenteric microvasculature, not mesenteric, this is premesenteric, sorry. And you can see that there is attachment of platelets to this, and this has not been manipulated in any way. The only inflammatory stimulus was that a distant myocardial infarction. For another study that we looked at was here in, this is to look at what happens with treatment with ponatinib. Ponatinib is used in hematologic malignancies. It's a tyrosine kinase inhibitor, and its use has been associated with an increased risk for cardiovascular events in patients. So these are mice that were treated for a week with ponatinib and then looked for whether or not they had platelet and VWF signals. And you can see that ponatinib seems to induce that in the microvasculature. These signals can be removed by the platelet signal and the VWF signal can both be removed by treatment with human ADAMTS13 or with N-acetylcysteine, which can remove von Willebrand factor from the vascular surface through disulfide reduction. Down here you have echocardiogram, and you can see that there's vascular wall motion abnormality, and that's accompanied by microvascular perfusion defects, which are shown here in these dark areas where there's no perfusion. But these defects are actually a consequence of microvascular occlusion and not macrovascular occlusion, as the angiogram looks completely normal. So in addition, we've looked to see whether this phenomenon is also accompanies aortic stenosis. Of course, aortic stenosis is when usually occurs in as people age, so it occurs in older people, and the risk factors are similar to those for the development of atherosclerosis. And of course, what happens is that the aortic valve narrows and the left ventricle of the heart has a much harder time pushing across a smaller cross-sectional area. So what you see here is that there's a very high peak velocity going across that aortic valve, and what we see here is that as you have an LDL receptor knockout mouse, you get an increase in the aortic gradient. But if that is also knocked out for ADAMTS13, that increase is even further. And one sees, and this is the peak aortic gradient, this is the mean aortic gradient. The valve resistance goes up with deficiency, with hyperlipidemia and deficiency of ADAMTS13. And just over here, this is the valvulo-arterial impedance, and this is an indication of total afterload on the left ventricle. And you can see that it's markedly increased, especially in the double knockout mice. So, in addition to doing that, we've done some staining, and this is a Mason trichrome stain, which here you have the wild type, the control animal. You can see these thin valve leaflets. There's no plaque in the aortic sinuses. Here you see in the LDL receptor knockout kind of a lot of accumulation of plaque and thickening of the valves, and that is worsened considerably by added ADAMTS13 deficiency. So you can see really a lot of plaque in here and quite thick valves in some instances. Staining for monocytes, here's the control. You can see that you have accumulation, but that increases in the double deficient mouse quite a bit. And of interest in both of these, you can see that those monocytes are accumulating in the plaque, but not so much in these thickened valves. So indicating that the increase in valve thickness is not just the accumulation of plaque there. This is picrocereus red stain, and this indicates matrix formation, in particular collagen. And you can see that yeah, you get an increase with LDL receptor deficiency, and even further within the leaflets, which you see very little here of the valves, increased matrix formation. Oops. Okay, and this is a stain here for von Willebrand factor. You can see in the control, you have it right in the endothelial layer. In the LDL receptor knockout, now you're starting to see it increasing in thickness. And in the double knockout, you have it not only in the plaque, you also have VWF accumulating on the valve itself. And that's the total valve area is quantified here. MMP9, matrix metalloprotease 9, that's produced by interstitial cells. You see very little in the wild type. It increases in the LDL receptor knockout, and you see massive quantities in the double knockout. So we think that this is a consequence of VWF on that surface that captures platelets that then release many growth factors. So I just want to say a few things about another phenomenon that we're observing, because people have talked about this, and this is looking at the accumulation on those pillars again of VW. Oops. This is looking at VWF accumulation here in citrated plasma and here in the same plasma that has been moderately recalcified. And you can see there you're seeing VWF accumulation. And then you get this, and this crazy stuff is so heavy that it actually is bending this pillar. And that we think is fibrin. And you can see that compared to in this plasma, the citrated plasma, the strands are long, but not as long as this thing, which is essentially extends all the way through to the end of the channel. That is not just blood clotting because we added back calcium, because it actually requires shear stress, because if we just leave in a tube, we leave that plasma static, it takes at least at 37 degrees, at least an hour, and at room temperature, 2 hours to clot. So, and that's fibrin. We can tell by this FITSI labeled polystat, which is polymer with fibrin binding peptides. The control polymer doesn't show that. And if we're going to have clotting there, you think you would have to have phosphatidylserine. So we tried to detect phosphatidylserine with lactadherin, and you can see that lactadherin, and this is in citrated plasma and a VWF strand. Those VWF strands appear to be capturing phosphatidylserine. And of interest, this stimulation of VWF activation appears to be a consequence of fibrin formation, is a consequence of contact activation of coagulation, because it can be inhibited with PPAC and also with contrypsin inhibitor. So to summarize, I've showed you that the ability of this surface-bound von Willebrand factor to capture platelets depends on its ability to self-associate into thicker fibers and strands. Self-association increases with shear and elongational flow, and it increases the ability of VWF to bind platelets. It's inhibited by HDL and ApoA1 and accelerated by LDL. And this endothelial VWF seems to be involved in many important human pathologies, including atherosclerosis, ponatinib-associated microangiopathy, and aortic stenosis, and of course, probably all of the microangiopathies. These VWF strands are interesting in that they can activate the contact pathway of coagulation and nucleate fibrin formation at high shear stress. So just to acknowledge everybody who's done this work, in particular, I want to mention Dominic Chung, who I've worked with now many years, and Junmei Chen. We basically, even though we're two labs, we more or less function like one lab. And all of the people in the lab that have been working on this, and of course the self-association stuff started way back with working with Jingfei Dong. Kimsey Platten, who's in the lab, has done all the recent work on that microfluidic device. We work with Ying, in particular on the vessels, with Jay and Tomas on the HDL, and with Jonathan and all of his group at OHSU. This has been a wonderful collaboration, and also with Sergio Fazio and Natalie Premier, who have helped us with a hyperlipidemia. So I will be happy to take questions.
Thank you very much, Jose. Dr. Conway, do you have a first question?
Yeah. Jose, thanks so much. I love it that you showed a picture of me from 1985.
Such a handsome man.
Yeah, I was, maybe. The microangiopathic, the schistocytosis that you see in TTP versus, say, atypical HUS. Do you think the mechanisms are, I know that you've shown this before with these strings and platelets and suggesting that there's fragmentation, hemolysis as a consequence of that. Do you think that the complement-mediated diseases like atypical HUS are the same mechanism? People always ask me about fragmentation. What's the mechanism?
Well, you know, what I'll say is that, I mean, here's my bias, that in those diseases, there is VWF on the surface, that you're getting this exact phenomenon. It's just that it's not a consequence of ADAMTS13 deficiency. It's a consequence of something else. Maybe it's a consequence of overwhelming the available ADAMTS13 because you stimulate so much VWF secretion. It's also possible that it's because the VWF or the ADAMTS13 become oxidized. So I think that if you really look at those vessels, if you really could travel in there in a little boat, you would see a lot of von Willebrand factor. That's what I think, but I may be wrong.
Thank you. Dr. Adili?
Oh, yeah, that was a very nice presentation, Dr. López. I'm just asking, how's the VWF able to bind the endothelial surface in the first place? And in terms of inhibition of platelet interaction to VWF, can you comment on targeting A1 domain versus GP1B? Which one is a better strategy?
Okay. Yeah, well, I can't really answer either question. I just will say that you look like you're on the Starship Enterprise. How is VWF attached? That's a great question. We thought we had figured it out years ago when we published a paper that said that p-selectin was the way that VWF was attached. But apparently, other people have said, even if you have p-selectin knockout, you still have it there. So I don't know. That's a good question, and one that I wish I had a much larger lab, because I would put a lot of effort into trying to figure out what that is. As to whether or not GP1B or VWF-A1 are a better target, well, I think there's arguments for both. VWF-A1, you're not going to activate anything if you block it, but there's a lot of VWF, right? Potentially a lot of VWF. GP1B, there seems to, certain antibodies that bind GP1B alpha will activate platelets, others will do strange things to megakaryocytes, and so there have been antibodies described that cause a quite marked thrombocytopenia. So probably VWF is a better target.
Thank you.
Thanks. Dr. Rand?
Thank you. Great talk. I may have missed this in your prior work possibly, but I was just wondering whether you've ever dissociated the VWF from these chambers or devices, and with either chaotropic agents or detergents, and if so, what are you seeing? Do you see the full range of multimers? Are you seeing any evidence of cross-linkage? Those are the kinds of things I was thinking about as I was seeing the VWF aggregate in these wonderful images.
Well, thank you for the question. We've talked about it, and in fact, we've just talked about getting it out and doing mass spec and seeing what else is there. We haven't really considered this idea of cross-linking. I think it's possible that in some cases it is, but I think it's probably one of these time-dependent things, because the initial, and there could be cross-linking in a number of ways. My colleague Jingfei Dong described that there was disulfide linkage, but it's possible also that there are, say, isopeptide bonds or other things like that. So I don't know the answer, but that's really a good question. We keep on talking about doing it, and for some reason there's always some technical glitch that we haven't done it. But thank you for the suggestion. We probably will go back and think about doing that again.
Thank you. Dr. Nee, I would like you to please put your question in the chat box, because we're a little bit out of time and running late. So, but
Yeah, no problem.
okay, thanks, Heyu.
Yep.
So our last speaker is Dr. Alan Mast, and Dr. Mast is a senior investigator at the Blood Research Institute of the Blood Center of Wisconsin, where he holds the Walter Schroeder Endowed Chair for Blood Research. Dr. Mast did most of his training, his MD and his PhD, both at Duke University and then a residency at Washington University in St. Louis. And Dr. Mast is going to talk to us today about work he's been doing on TFPI. Alan, over to you.
Great, thank you so much. Let me see if this works.
It should work for you to just share screen.
Yeah. Let's see what happens.
Yeah, something's happening. There you go.
There. Is that the wrong screen now, right? Is that right? Okay.
Looks good.
Yeah, we can see your slides.
Okay. So thank you so much. So Ed, thank you for inviting me to present at this. It's a truly an honor. If I think of all the things I've done in my career, being invited to give talk by you at your retirement is way up there on the list. I truly appreciate it.
Alan, you need to switch your display.
It's backwards now?
Yeah.
Oh.
There you go.
Does that work?
Perfect.
Okay. Oops. Anyhow, so just a brief introduction. I've been fortunate to be able to work with Ed on his CD248 project that just came out in a very small way, and I've enjoyed that. And in addition, mostly Ed, I've just enjoyed getting to know you as a person and as a friend, and having time to talk to each other about science or about other things as well. And of course, we all miss those times. And I keep saying it's gonna be less than a year. Used to say it's at least a year. We'll be actually, it's gonna be less than a year till that happens. So I guess today what I want to talk about is the work we've been doing, looking at, we found that TFPI deficient mice have a severe vascular effect, and that's shown on this slide here, where this is a picture of the brain, and these are blood vessels entering the brain, and they're not normal. And I'm going to describe what we know about this and see if we can get people here to help me understand it better. So just for an outline to talk, first I'll give you a background of TFPI biology. TFPI is sort of a complicated protein for people who don't study it all the time to remember. And so I want to review it so people can help try and understand what's going on. And then I'll present this vascular phenotype, and this will be most of the presentation. And the slides shown here, again, show in more detail what was on the title slide. This is the brain of the mouse. These are the meninges right here. And this is a normal mouse. And you can see that normal vascular development in the brain happens by angiogenesis, where blood vessels dive in from the surface of the brain, the meninges, into the brain. You can see this is a slide that just has a really long section of a normal blood vessel. And you can see there's other blood vessels along here diving into the brain normally. And this contrasts when you have the TFPI null mouse. This does not happen, and you get these really odd looking structures. And so this is during embryogenesis that this happens. And so we want to use all the knowledge that we've spent trying to figure out about TFPI biology over the years, and can we understand why this vascular phenotype is happening in the mice? I think it's really important because to me, this is one of the, and I'm talking about my own work, so you guys can say I'm over importantizing it, but it's one of the best examples, I think, of blood coagulation proteases being directly involved in vascular development. And so, that's right. So to start about talking about TFPI, TFPI is an alternatively spliced protein, which makes it interesting and complicated to understand. So this protein here is called TFPI alpha, and it's this triple-headed Kunitz type protease inhibitor. And the second Kunitz domain directly inhibits factor 10A. And in the presence of factor 10A, TFPI is a very good inhibitor of tissue factor 7A. And this is classically what TFPI was described as. It's the best, and I think the only physiologically relevant inhibitor that's been described so far of the 7A tissue factor complex. It's also been described over time that this third Kunitz domain binds to protein S, and that might help TFPI alpha bind to cell surfaces and enhance its ability to inhibit factor 10A. And then importantly for this talk is the C-terminal region of TFPI is very basic, but it also has homology with factor 5 and allows TFPI alpha to be an inhibitor of early forms of prothrombinase. So TFPI alpha is found in plasma in humans and in platelets, and is so it's considered a soluble protein. It's also considered a heparin releasable protein. So when you infuse heparin into a human being, their plasma TFPI alpha levels will go up 2 to 4-fold. A second form of TFPI is called TFPI beta, and it's a truncated form of TFPI alpha. It only contains the first 2 Kunitz domains, so it's a very effective inhibitor of 7A tissue factor, but it doesn't have the factor V binding sequence, so it doesn't inhibit prothrombinase. Instead, what it has is this GPI anchor attachment site, and that allows it to attach to the cell surface. And so TFPI beta is found on endothelium and on monocytes. All right. So I want to go back briefly here to talk about TFPI alpha for a minute. I mentioned TFPI alpha is a heparin releasable protein, and it's always been the dogma around TFPI alpha is that it's binding to glycosaminoglycans on the surface of endothelium. So the dogma is that TFPI alpha and TFPI beta are both found in the same location on the endothelium surface, just attached in different ways, where TFPI beta is attached to its GPI anchor, and TFPI alpha is binding more non-specifically with glycosaminoglycans through this very basic region. And I wanted to present just in one slide some very recent work that Julie Peterson in my lab has done. And we found that the main pool, the major reservoir of heparin releasable TFPI alpha is not the cell surface. It's not on the endothelium. Instead, it's in the extracellular matrix. And this paper just came out in press in ATVB. And these are really cool pictures that I like a lot. And this, to me, this, if you say a picture is worth 1,000 words, or this picture is sort of worth 4,000 words, you don't have to read the whole manuscript. You can just look at these pictures. And what they are is they are, these are just ECV 304, EAHY926 cells. They're EAHY926 cells are of a recombinant endothelial cell line in culture, and then the nucleus is stained with DAPI, and TFPI alpha is stained red. And you can see that the nucleus, and then underneath the cell is all the TFPI alpha. And when you sort of can flip these images around, you can look at in different angles and see, again, there's TFPI alphas all in extracellular matrix. It really shows best here in this bottom picture, where you can actually see the contour of the extracellular matrix being laid down by the cells, and TFPI alpha just all throughout it. Also really interesting in these cells is that TFPI alpha is found in an intracellular granule within these cells. This was originally reported in 1995 by Floria Lupu, and no one has ever characterized these granules other than that original paper by him, where he said there's a granule and distinct from libopilati bodies. So I think there's a lot more work to do to try and understand TFPI alpha in these granules as well. But nevertheless, and if you read the paper, we show in pretty good detail that the large majority of TFPI alpha, both in cell culture and in vivo, comes from the extracellular matrix, not from the endothelium, which sort of changes how you think where TFPI is and how it regulates tissue factor activity. Okay, so we've spent a lot of our time trying to understand how TFPI binds to factor V and how it inhibits prothrombinase, and review that just briefly, because it's important for some of the studies I'll show you with the vascular development. So this is, we recognized a long time ago that TFPI has a homologous region and it's C-terminus with factor V. And there's lots of proteins that have the long stretches of basic amino acids. But what's unique for factor V and TFPI is they have this LIT as well as combined with the stretch of basic amino acids. And that's not, that's first, it's well conserved in all mammals, and it's no other protein in mammals have this sequence. All right. And this sequence is what mediates the inhibition of early forms of prothrombinase by TFPI. When I say early forms of prothrombinase, I mean forms of prothrombinase that assemble before thrombin is generated. So 10A, 5A coming together, but they haven't cleaved prothrombin to thrombin yet. Okay, so this is how this happens. This is just the structure of factor V. Has this large B domain in the center of the protein, and this is just an activation peptide that's over 700 amino acids long. And what Mateen Boss and Rodney Kumari showed is that there's a basic region and an acidic region that help factor V fold into its cofactor conformation. And if you remove either the basic region or the acidic region of factor V B domain, you'll activate factor V to an active clotting factor. And this is the section of TF that has homology with TFPI. So this is the region of the B domain. So it's sort of hard to understand why does TFPI having homology with it, just part of the activation peptide, have any relevance to biology? And that's Jeremy Wood, fortunately came into my lab and really helped figure this out. And he worked with Paula Tracy and knew that there were different forms of factor V A that could be formed, like when factor 10A cleaves factor V, you get a form that looks like this. It has the B domain, but it's missing the basic region, but still has the acidic region. And there's also forms in platelets that are similar to that. And this last form on the bottom is when thrombin comes and totally cleaves the B domain. And so what, to make a long story short, is what happens is TFPI alpha, it binds extremely tightly to these forms of factor V and allows it to inhibit prothrombinase. And this is important. Now I'm going to go through a couple cases of why it's important in bleeding and clotting. And at the same time we were doing the study showing TFPI has homology to the B domain and inhibits prothrombinase, Bjorn Dahlbeck was figuring out this form of bleeding disorder called factor V East Texas, which is a moderately severe bleeding disorder. And these patients, they have a mutation in the B domain of factor V that allows them, that causes alternative splicing in the factor V B domain, such they make this form of factor V called factor V short, which is very similar to the forms I showed you on the last slide. And these patients have high levels of circulating factor V short that bind tightly to TFPI. So instead of having an active clotting factor, they actually have an anticoagulant complex of TFPI and factor V floating in the plasma. And this results in them having really high levels of plasma TFPI alpha. And we think this is a circulating prothrombinase inhibitor that contributes to the bleeding disorder. So another thing that was really fascinating is you start looking for where you see this LIKT followed by the basic region, and we saw it. There's a black fly that lives in northern United States, I think, and sort of bites people and sucks their blood. And so people have studied their whole salivome of these from these flies, and one of the proteins they pulled out was, we called it black fly protease inhibitor. It's described in this paper, and it's an anticoagulant protein, and they screened it against a whole bunch of proteases, and it was the best at inhibiting factor 10A. And this is the structure of it. It's a single Kunitz domain linked to a basic C-terminal tail. This is almost identical to TFPI alpha. There's this LIKT region and the long basic region. And for some reason, it has a proline in here. We don't know why. But it just, to me, this is just fascinating because of fly saliva and humans have, I guess that's convergent evolution. We've come upon the same idea as a way to inhibit clotting. And to me, finding this just showed, wow, it's just intellectually satisfying. It makes me feel like we're not just following a red herring, or it's something that's interesting but not physiologically relevant. To me, this really encourages me that this is really something that's important and is happening in our bodies. So what I think these data have shown is that there's an important regulatory point of coagulation is when the factor V B domain has been cleaved by 10A, but before thrombin is generated. When I was originally talking about this one night at dinner with Ken Mann, it was all brand new in my mind. He was going, this will never happen. Once thrombin is generated, it takes 2 nanoseconds for all the factor V to be totally completely cleaved to factor Va without this B domain. And he was a little skeptical of it. And I think it is an important, because of what I've showed you so far, I think this is an important region, and these forms of factor V that are generated before thrombin is generated are important. And so lastly, in that's sort of the background, we've also, we're really interested in, could this be involved in thrombotic disorders as well as bleeding disorders? And I've always been intrigued by this work by Dan Eitzman and Randy Westrup, where they show that mice with factor V Leiden and TFPI haploinsufficiency have severe perinatal thrombosis. And so what it was, is you just have to take a factor V Leiden mouse and take away half of their TFPI, and they would clot to death before they were born. And I always thought this might be something more than just two mild procoagulant states being additive. There's something synergistic going on here. And so this was another project that I had Jeremy worked on when he was in the lab. And just to summarize it really quickly, using thrombin generation curves, we think that there's two reasons that factor V Leiden is prothrombotic. And if you look here, there's a thrombin generation curve with factor V Leiden and a thrombin generation curve with normal factor V. And if you, the APC degradation of factor V is delayed or not as good when you have factor V Leiden, and that allows there to be more thrombin generated during the propagation phase of clotting. We also showed in this paper that we think that factor V Leiden, when prothrombinase is assembled with factor V Leiden, it's not inhibited by TFPI alpha as well as regular prothrombinase. We think it's because of how this LIKT region interacts with parts of factor V and it doesn't interact well with factor V Leiden as well as it does with normal factor V. Okay, so that's the background of TFPI biology, and now we'll go to I'll summarize it. So the summary of TFPI alpha is platelets are the only make TFPI alpha. There's no TFPI beta in a platelet. Heparin releasable TFPI alpha appears to be an extracellular matrix. TFPI alpha inhibits early forms of prothrombinase, and that's before thrombin is generated. And inhibition of early forms of prothrombinase by TFPI appears to be physiologically relevant, and is showed by insect bites and bleeding disorder with factor V textus and with the clotting disorders with factor V Leiden. Okay, so now we'll go to cerebrovascular development. So what this is, I'll talk about with late stage gestation of TFPI embryos that are deficient in the first Kunitz domain of TFPI. So these TFPI K1 null embryos, they die during embryogenesis, and they die in two stages. There's about half of them die during early or mid gestation, which is between days 9 and 11. But the ones that make it through that stage tend to hang on and die variably between day 12 and 18. And no TFPI null embryos have ever survived. Occasionally we have some born, but they're dead when they're born or die very shortly after birth. And so this is the stage we want to look at today. What's happening to these guys? Why are they dying? And so we had a lot of clues that there was problems with the brain. And so Susan Maroney in my lab, I'm really fortunate to have her work with me. She's a DVM PhD who did training in veterinary pathology. And so she is very talented at looking at slides and studies them and learns things. And she really figured all of this phenotype out. And I'm showing first here, this is just a hematoxylin and eosin stain of a wild type mouse brain, and then a TFPI null brain. And at first look, they don't look that different. And I think that's why this was not characterized initially. The TFPI null mice, I think, were originally published in 1997, and this was not recognized. And so we figured this out more recently. And you can see some things in the TFPI null mice. Their brains seem to be a little smaller. They're not totally filling the calvarium. And there's also these little red spots in here occasionally that were noted in the original paper that was saying maybe there's fibrin deposits in the brains of the null mice, but that's all essentially was described. And so what Susan did is she stained the mice, stained these brains with different stain called the Martia scarlet blue stain that really brings out the phenotype. And what you can see here is the outline, this dotted line is an area of brain tissue that's dead and has the yellow cells in here are red blood cells. And then you can also see that sort of surrounding is highlighted by the areas are these little dots or whatever. And we're trying to figure out what these were. At first we thought they were just blood clots and we didn't know. And so we started studying them more. And what we found was if you stain them with laminin, it would stain the basement membrane of these vascular structures that are not normal in these TFPI null mice. Okay, so we started looking at these were, and just characterizing how prevalent are they and how often do we see them. We saw them in 12 of 12 TFPI embryos from 7 litters between E12.5 and 16.5. And they were not present in 50 of 50 TFPI positive or TFPI heterozygous litter mates from these litters. And so what this means is this is a deletion of the K1 domain produces a complete penetrance of this vascular phenotype in TFPI null embryos. It's actually better than genotyping. There was a while that we thought this was wrong, and we were seeing this in heterozygous mice. And it was turned out that our genotyping was messed up, not that the phenotype was messed up. So we went back and figured that out. So what these are is these are glomeruloid bodies, and I'll tell you what a glomeruloid body is. This is just a normal vessel stained in a wild type mouse, and green is thrombomodulin, and yellow is lamina. You can see they have nice, well-defined structures. When you do the same stain on a glomerulus, one of these structures, you get something that looks like this, and you can just see there's these multilobular vascular with endothelial cell disarray. These endothelial cells are not forming normal vessels and the TFPI null mice, and they're called glomeruloid bodies. These are called glomeruloid bodies. They're also seen as a diagnostic feature in patients with brain cancer. But these mice don't have cancer. This is just a developmental effect. And they're called glomeruloid bodies because they resemble the glomeruli of kidneys. And that's why they're called glomerular bodies. So one of the interesting things, there's other knockout mice that I'll mention a little bit that have been described that develop glomerular bodies as well. And they've been described as affecting specifically only hindbrain development or forebrain development. And so one of the things we did is we did coronal sections through the mice. And you can see, and stain them with laminin here, and you can see that these glomerular bodies form throughout all sections of the brain in the TFPI null mice. So they're everywhere. And they also form in the spinal cord, and you can see them sort of in different places. This is a place in the spinal cord where we found a lot of them. And what's interesting is earlier, Paul Ellery, who worked in the lab, published a paper where he showed that PAR4 null mice would rescue some of the TFPI K1 null by all the way to adulthood. But some of these mice were born with short tails. Not all of them, but some of them, about a third of them had short tails, and they were always born with whatever tail they maintained throughout their entire life. So if a mouse was born with a long tail, it kept a long tail through its whole life. And if it was born with a short tail, it had a short tail. Because the reason I say that is because this doesn't seem to be a thrombotic event. It's not something where they clot off their tail because they don't have TFPI. And so what we hypothesized is that these glomeruli bodies, which form variably, can affect tail development in these mice. And the ones that have a lot of glomeruli bodies forming in the right place at a spinal cord might still survive but have a short tail. And we're working more to try and prove this. This is sort of a difficult thing to prove, but I think it's an interesting hypothesis. And the other thing that's really interesting here is that all the other vasculature in the mice is normal. So this is a cerebrovascular specific defect. And what's interesting, and I'm just learning all this stuff, is that the brain vascular develops by angiogenesis, whereas the other vessels in the body develop by vasculogenesis. So it's a different process, and it seems like TFPI is specific for affecting how angiogenesis happens during development. So we had done a lot of work with TFPI and factor V and prothrombinase, and so we wanted to see, does altering factor V modulate this phenotype? And so this is where we started working with Audrey Claren, Randy Westrick, and David Ginsburg, crossing them into the factor V null mice. And what we found is you take a TFPI K1 null mouse and a factor V heterozygous mouse, the phenotype is totally unaltered. This is severe hemorrhage in the brain, and this part on this side of the dotted line, the brain is dying, and there's all these glomerular bodies. What was fascinating is when you totally got rid of factor V, the phenotype totally disappears. So you go from this incredibly sick brain to this totally normal brain just by getting rid of factor V. And we went through and did a whole bunch of litters and had Susan read them in a blinded fashion, and of 8 litters with 53 embryos, there were 5 TFPI-K1 null, factor 5 null, and all 5 she read as having normal brain histology, not knowing what these were. Okay. So from these data, we think that TFPI modulates thrombin production in a manner that substantially alters vascular development in the CNS. Because we can knock out factor 5 and restore the phenotype, there's other places it could be happening, but I think it's mainly by modulating thrombin production that TFPI is doing this. So now it's where we get to a little bit of the speculation about what could be happening. You automatically, we think of PARs in embryonic development. So TFPI is modulating thrombin production, which modulates how PARs are being cleaved during vascular development in the brain, and that's the phenotype. And there's some evidence this could be true. Sean Coughlin wrote a review about vascular development in PAR1, and it says, PAR1 knockout has partial mid-gestational lethality with bleeding that may be secondary to endothelial defects rather than platelet defects, which would be consistent with what we're seeing. And modulation of thrombin generation by TFPI may alter PAR-dependent vascular development. That's certainly one hypothesis we have, but I want to show you that I don't think that there might be a better hypothesis. And we came across this paper from Ben Barr's lab that was said that Wnt beta-catenin signaling is required for CNS, but not non-CNS angiogenesis. And I said, well, that's exactly what we're seeing in the TFPI mice. We should probably look at this. And one quote from this paper, or I think it's from the abstract, is that these defects include reduction in vessel number, loss of capillary beds, and the formation of hemorrhagic vascular malformations that remain adherent to the meninges. And that's what I showed you exactly at the very beginning of the talk. Those were all the vascular malformations that were adherent to the meninges. So to me, it seems like we have this very similar phenotype that's converging between TFPI and Wnt signaling, and we're trying to put this together if it's true. And so just a little bit more about beta-catenin signaling. There's two different ways that it happens. There's some that affect forebrain and spinal cord angiogenesis, and then some that affect hindbrain and spinal cord angiogenesis. And that's why I mentioned that TFPI affects all angiogenesis in the brain. So somehow it might be affecting both of these beta-catenin signaling pathways. But if you just take one of these proteins in here, it's called GPR124, and if you knock it out, they get glomerular bodies, and it's just, I put this figure that I showed you earlier in the talk. This is TFPI, and it's glomerular bodies along the meninges, and this is a figure from that paper showing the glomerular bodies finding along the meninges, just very similar phenotypes. And so we're doing, once you can hopefully appreciate when we do all this, there's so many things you can try and do to figure out what's going on. And we're trying to sort of step by step, as we can develop the mice and study these things, figure out how the clotting pathway is directly or so dramatically affecting vascular development in the mice. And so one of our hypotheses is, instead of going through PAR signaling, maybe thrombin, there's too much thrombin being developed in the TFPI null mice, and it's degrading some of these beta-catenin signaling components. But we have not been able to prove that. We've done some experiments, and it's just a hypothesis. So just to summarize this, TFPI-K1 null embryos develop vascular anomalies called glomerular bodies. And they occur in the brain and the spinal cord, but not in other tissues. And glomerular bodies are associated with hemorrhage, cellular death, and they likely contribute to the late gestational death of these mice. There are people that ask me sometimes, well, a mouse can develop without a brain. Why do you think this is killing the mouse? And I think, well, a dead absence of a brain is different from a very sick brain. And so these mice have a really sick brain, and I think that's what's killing them. It's worse than having no brain at all. That's the hypothesis. And then glomerular bodies do not form in TFPI-K1 embryos lacking factor 5, implicating TFPI-mediated control of thrombin production and cerebrovascular development. And again, this is why there's lots of reasons I'm excited about this project, but I think it's linking the blood coagulation pathway directly to vascular development in a way that hasn't been described before. And then finally, I mentioned this alteration of beta-catenin and PAR signaling pathways. And so just to summarize, this slide shows the people who did the work and highlight Susan Maroney, who's worked with me for many, many years and really figured out the glomeruli body project. And then all the collaborators who helped along the way. So with that, I'll stop. Take questions.
Thank you very much, Alan. It's really interesting. We're looking for questions. If you could stop sharing your screen, then we'll get the full Hollywood Squares view, figure out who's doing what.
Okay.
Okay, great. Ed, unmute yourself.
Yep. Couldn't go a whole day without being told to unmute. Alan, thanks so much. There's so much to cover there from development and then before. I'll just ask like a couple of questions. One, is TFPI alpha from platelets? Is it in the microvesicles? In regards to Kelly's talk. And then second, the specificity of TFPI. There's data that shows that it interferes with MASPs, the MBL-associated serine protease 2, I think, and thus interferes with complement. And it's interesting that you brought up the issue of saliva from flies, which is obviously complement plays a major role there as well. I'm just curious how much information there is about TFPI in complement or other substrates for that matter.
So I specifically went back and sort of reviewed the TFPI Mast paper for this presentation because I do remember at ISTH you brought that paper to my attention, and I don't know. The part of the paper that I'm concerned about is that it has it's the affinity for TFPI K2 domain for Mast versus factor 10A is, I think, 4,000-fold less affinity. So you have to someplace you have to have a lot of TFPI to be an effective inhibitor of Mast. That's not saying that couldn't be true. We have all this TFPI now in the extracellular matrix, and I don't know how much is really there.
Right.
So it could be, and it's certainly worth pursuing. We have other projects where we're seeing, TFPI could be affecting fibrosis, and platelet TFPI affecting fibrosis. No, I have not talked to Kelly. I don't know if Kelly's looked for TFPI in her
I have not, but we're doing comprehensive proteomics, so you will be the first to know if it's there.
Great. Okay, so stay tuned.
Stay tuned.
And just to follow up on your embryology, just to clarify, these TFPI Pringle 1 factor 5 null, are those just embryos? Have you looked postnatally, or do they survive?
Right. So they don't. If they'll bleed to death though. So without factor 5, they'll be born and bleed to death. And what we can do, and this is work that been done by Audrey and Mandy and Dave Ginsburg, is they have the transgenic factor 5 mice. So if you just have a little bit of TFPI in your platelets, a factor 5 in the platelets, then you can get a TFPI null, factor 5 null with transgenic a little bit. And those mice live, they survive. And we're working on hopefully trying to publish the phenotype of those surviving mice.
Alan, there's a question from Dave Farrell in the chat box asking, were there any similarities in the TFPI mice to the prothrombin knockout mice, which he thought had defective blood vessel formation?
Yes, I do not know, Dave. I'll look. Not that I remember seeing or it's come across me, but I haven't. I need to read about that.
Okay.
Thank you for asking too.
Dr. Prysdale, you've come on with your camera. I'm assuming you have a question.
I just want to say hi. Yeah, I do have a question. Alan, lucid talk as always. Nice, slow delivery. I love how you speak. I was wondering, platelet factor 5 has always been considered pre-activated. If it's sitting there in the presence of TFPI, presumably it can bind to the TFPI. Is that because it's not saturated with the TFPI, or because it's not, factor 5 is not there in high amounts in the alpha granules. What's the deal?
Right. So that's an interesting question and something we thought about. What's important to know is that TFPI and factor V are not in the same location in platelets.
Ah, I didn't know that.
TFPI is not in alpha granules. Now, I don't know why it's not in alpha granules, but it's clearly not. And we did a big study of a whole bunch of blood donors looking at their factor V and TFPI levels and platelets, and they don't correlate well at all, which very much contrasts with platelet TFPI and factor V levels, which do correlate, because TFPI will alpha interacts with even the normal forms of factor V to some degree in plasma.
So where is it? It's cytosolic?
Factor V short is this main interaction. Yeah, what?
It's cytosolic? Or
Yeah, we think so.
huh,
Yep.
interesting.
It fits with my theory that there's nothing easy about TFPI. I would say Guy could have just put it in alpha granules and then we just feel so it's an alpha granule, but it's not in alpha granules. And where it is not in alpha granules is, is there a second granule that hasn't been well characterized or another granule that has TFPI, maybe some other proteins in it? We haven't been able to show that, but it's clearly not in alpha granules.
Thank you.
That's interesting because C1 inhibitor is also being described as being cytosolic and not
Yeah.
in alpha granules.
And so is factor
Other
13.
inhibitor, yeah.
Yeah. It's not unique.
And factor 13A chain.
So Ed, how they get, when the platelet gets activated, TFPI gets released, and how that TFPI then interacts with factor V and how fast it happens and what the kinetics of that is are all, it's not known.
Well, you heard Ken, nanoseconds, man.
Nanoseconds to activate all the thrombin. Once thrombin's generated and that factor V domain gets totally cleaved, TFPI has no binding at all to factor V and the reaction's over. So this has to happen only really early in a procoagulant response.
Interesting. Thank you.
Other questions or comments? Okay, well, hearing none, I would like to, first of all, just on behalf of the Center for Blood Research, thank all of the speakers for being willing to participate in this symposium. It was a really great few hours and I've learned a ton, which is always fun. And I thought I might give our guest of honor, Dr. Conway, the last word.
Okay, well, thank you so much. I won't keep you long. Let me say a few words. All right, so first to start, thanks to Dana, Mira, Stephanie, Christine for organizing this amazing symposium. And then of course the speakers, Alyssa, Kelly, Owen, Patricia, Jose, and Alan. Really the best of the best. Thanks for taking the time to be here, present your fabulous work. And then to everyone who's come, just imagine in non-COVID times, we could have had one fantastic party. So although my name is at the top of this program, this is really a testament to the efforts first of Dana, Ross, Don, Chip, Grant, and others who created the Center for Blood Research. And then everybody who participates, you built a place, a center to be proud of, and that's flourished and continued to grow. A little history, in 2007, I met Ross over beers, what else, at ISTH in Geneva. And it was there that we first started to discuss my return from Belgium to Vancouver. And like all recruitments, it didn't go exactly smoothly, but particularly Ross and Mike Barnett and Grant at the time and others pushed things along, so it happened. It was a few months just before I was slated to come that Ross mentioned the CBR director position. I wasn't coming for that particular. I thought, okay, sort of cool. Why not? Something new. I had no idea what was involved. It sounded interesting. There was great science. It was multidisciplinary. There was apparently lots of resources. A new building had gone up a few years before. They had a training program, mentoring programs. They had money. They had links to Heart and Stroke Foundation, Michael Smith Foundation. So July 2009, I arrived in Vancouver. For those who may recall, the recession had just hit, wiped out pretty well all of CBR's financial reserves. All the support for training programs disappeared. I had to cut positions in core facilities. And really, I don't think I was so popular in those days. And that was my start. But with the help of a fabulous steering committee, which included Dana and Ed, who are here, and others, Chris Orvig was there, enthusiastic membership, I donned a fundraising cap. I'd never fundraised before in my life. Learning along the way, partnering with some wonderful development people. Pharma contributed, and over the years, we got lucky with some generous donors. And as the pennies rolled in, we gave it out just as fast to support students and trainees, really the bedrock of all that goes on here. And then we hired some young and creative staff, the likes of Christine and Stephanie and Mira, to build and run education programs. And then Ross's initiation, of course, of the Earl Davey Symposium really helped us put us on the international map. And before you knew it, we had a CBR that looked, I think, like the vision that Ross and Dana and Dawn and the other founders had years before. So as you know, there are several world-class centers for blood research. Many of you hail from those. And I think ours is particularly unique and special, at least to us, because of the community that we've built. And it's not just the science, although that's obviously the cornerstone of our existence, but it's also everything that's important in this sort of crazy world. So, and it's also what makes it so much fun to be here. So I know that Dana will lead the CBR in new and creative directions. And my thanks once again to all of you. I wish you all to stay safe. And I look forward to seeing you in the near future, like in real person. So thanks, Dana, and everybody.
Thank you very much, Ed. And take care, everyone. I think this brings us to a close.